Well, good morning, everyone. It's the first Wednesday in April, so I thought the tulip background would be appropriate. But today it's my pleasure to introduce Dr. Abner Louissaint, who is a hematopathologist at the Massachusetts General Hospital. Dr. Louissaint grew up in New York City, or in the New York City area, but then moved to Boston to complete residency and fellowship and postdoctoral training at the Mass Gen Hospital. So Dr. Louissaint and I knew of each other. I think that he was always highly regarded by a mutual colleague and also friend, Dr. Elizabeth Corville, but we never had a chance to work together until recently. He really helped me out with the case. So one of the things that Dr. Louissaint has been able to do is to really kind of characterize hard to classify B-cell lymphomas. And so for me, with my case, what was difficult about it is it was a patient that had ultimately a follicular lymphoma, but it didn't have the kind of the classic genetic background that we'd expect. And I think Abner helped me put the pieces of the puzzle together to really get the right diagnosis for the patient and also perhaps refute an opinion of some of the experts. And so it was really helpful. So Abner has ascended to the rank of associate professor as an endowed professor now. And some of the cool stuff that's going on is that he's created these really neat xenograft models. So if you think about B-cell lymphomas, and he's looking at kind of two different types, follicular lymphoma that doesn't have the classic 14/18 translocation, and ALK-positive large B-cell lymphoma, they're difficult to study because they're heterogeneous. And so he's created some xenograft models to take patient samples, put them in the models, and then do single cell analysis to look at intratumoral heterogeneity. So today he's going to talk to us about translating molecular characterization and preclinical investigation of B-cell lymphomas into impactful therapeutic opportunities. And Dr. Louissaint, I really thank you so much for sharing this information with us on our grand rounds, and welcome to Minnesota, albeit electronically.
Thank you so much. It's such a pleasure and honor to participate, and thank you for inviting me to speak. And, I have no disclosures. So a lot of what I've been focused on over the past couple decades really is looking at, hold on, looking at lymphoma classification as we do in pathology, but also leveraging our unique window into disease to really identify aspects of these cases and entities that impact therapeutic intervention and behavior to therapy. So, everyone relies on us for classification of lymphoma, but I think my philosophy has been we, especially at academic centers, we get to see a lot of unusual cases and a lot of cases in general. So we're able to observe unique features of cases, even within a particular entity, that if we leverage correctly, we can often potentially correlates with some molecular phenotype that could impact either the way the tumor behaves or the way the tumor responds to therapy. Um, and so that's the thing that links a lot of the work that we have done. And so it's really a lot of founded in the heterogeneity within defined lymphoma entities and defining biomarkers, not only for classification, but to predict therapeutic effective therapies and behaviors. So my work really started in the area of follicular lymphoma and unique follicular lymphoma subtypes. And to start, I just wanted to begin with a case of a 25-year-old man with isolated cervical lymphadenopathy, limited stage disease, was CD10 positive by flow cytometry. And basically there was a follicular pattern with and a high proliferation fraction, a lot of CD21 positive meshworks, which indicates the presence of follicles and mostly BCL2 negative with some BCL2 positive T cells interspersed. So at this point in time, based on the morphology, it was entertained, could this be grade 3 follicular lymphoma, which would lead to chemotherapy, right, as opposed to a low-grade follicular lymphoma, which would basically be observed or receive radiation therapy. And just as a background for those not as familiar, follicular lymphoma is a neoplasm of germinal center B cells within the center of the follicle. It grows, has a follicular growth pattern. It represents about 20% of all lymphomas, and patients usually present in the sixth decade in that area. Usually a pretty similar male to female ratio. Most cases present with advanced stage disease, often involving bone marrow, lymph nodes, and high-grade transformation occurs at a low but uh, steady rate. And while this has changed in the WHO, but it's still optional to grade follicular lymphoma. Based on a number of smaller cleaved irregular cells called centrocytes, or larger round cells called centroblasts, and essentially, traditionally, more than 15 or more centroblasts per high power field would qualify for a high grade or grade 3, and then whether those are sheets of large cells or just more than 15 per high power field, that determines grade 3A or 3B. And this the designation really impacts therapy. And follicular lymphoma is founded with one of the initial genetic alterations is BCL2 translocation, in which uh, uh, there's a juxtaposition of BCL2 to Ig heavy chain, causing overexpression of BCL2, which is anti-apoptotic and allows neoplastic cells to survive. We have a lot of tools in our toolbox to make a diagnosis of follicular lymphoma, including looking at BCL2 expression by immunohistochemistry. So because of the BCL2 overexpression, we can detect that in the germinal centers, whereas in a reactive node, that's typically negative. And these cells are co-expressing markers of the germinal center, or BCL6 and CD10. And typically, a normal germinal center has a very high proliferation fraction, but follicular lymphoma tends to have a very low proliferation fraction. So that is also a suggestion of lymphoma. In addition, we can assess for clonal IgH by PCR, as well as looking for clonal population by looking at a predominance of kappa versus lambda by flow cytometry, and we can actually look for the BCL2 rearrangement by FISH. So it's usually a pretty straightforward diagnosis. Now, a few decades ago, several decades ago, folks began to realize that there were unusual cases that occurred in children of follicular lymphoma, and these tended to occur in male patients. So there was several case series and some trends across those series. Young boys were much more involved. It occurred much more commonly in young boys than girls. They were limited stage. They usually were restricted to head and neck. Typically, they had what looked like a higher histologic grade, but they lacked BCL2. And many of these patients were treated as if they had high-grade follicular lymphoma, but they always did really well. And these patients had a durable, remission with chemo. By 2008, in the WHO, it was recognized as a provisional entity based on its unusual indolent behavior despite its high-grade morphology, but there were really no other features to distinguish it from follicular lymphoma that occurred in adults. So I got involved because as a fellow, I was beginning to see in consultation a lot of cases that were occurring in patients who were in 20, 30-year-olds. So not technically kids, but they had these follicular lymphomas, which was still unusually young. So it seemed to me to be important to characterize because they had a very different prognosis than a conventional high-grade follicular lymphoma. So the question became, what are the real parameters that define what was called pediatric follicular lymphoma? Can we define objective criteria? And so the first thing we did was we looked at our cohort at MGH of all patients with follicular lymphoma less than 40 years of age. We found 27 cases, and we found that there was a sort of a binary situation where the majority of patients had limited stage disease. About 43% of them were older than 18. Our cutoff was less than 40. And many of them, the majority of them, were male patients. whereas the other sort of group was the higher stage patients, basically with a 2 to 1, very similar male to female, uh, much lower male to female ratio, and um, obviously they were almost all, I mean, 80% of them were greater than 18 years of age. So it seems to be sort of an interesting difference there. And when we looked at other features, the stage ones tended to have what seemed to be if you just looked at size of cells, a higher grade uh, picture, more of a starry sky pattern, larger follicles. Um, so there were some morphological features that were different. Um, and then when we looked at progression, we noticed that the ones that were low stage really did not progress or relapse, whereas it was very common for the higher stage ones to progress or relapse. We also noticed that they didn't have, we had done FISH on all of these for BCL2, BCL6, and looked at MUM1. And all of these did not, the stage one ones did not have a BCL2 or BCL6 rearrangement, and they all had a very high proliferation fraction greater than 30%. And when you look, and obviously when you looked at both of those parameters, all of them had both of those. Whereas the higher stage cases, really none of them had both, and some of them had lacked rearrangements. Half of them had a high proliferation fraction, but again, the combination seemed to be unique to these lower stage cases. So we began to think that perhaps this was a different some different subset. We're beginning to identify some unique subset in follicular lymphoma that could maybe predict, these cases that had a different behavior. And so we then looked at an adult cohort, just a traditional adult cohort, and tried to look at cases that had no translocation and had of BCL2, BCL6, and had a high proliferation fraction. And there were about, out of 40 cases, there were about 8 of them. All of these were stage 1, versus the other groups where you had either translocation or high proliferation of both, no translocation, high proliferation of both, and these did not demonstrate um, uh, low stage uniformly. And in terms of progression relapse, we were surprised to find that this first group, which really had, defined by the lack of translocations and high proliferation fraction, really demonstrated no progression relapse. And interesting, when we looked at the notes, I remember seeing how many of them were sort of, the clinical notes were sort of really shocked and almost like describing miracles happening. But really, we began to think that there is something that actually we can hang our hat on that may, irrespective of the age, may predict the outcome patients. And this is just a Kaplan-Meier showing in terms of progression, showing the cases with no BCL2 arrangement, high proliferation fraction, and how there was no progression versus the others that had one or the other. So we sort of started to call these pediatric type follicular to recognize the fact that this entity, whatever this is, really seemed to occur in both, while it was mostly young adults and children, could occur in adults as well. At the same time, it began to recognize Elaine Jaffe's work that these cases of pediatric type follicular lymphoma really had a different morphology. So I mentioned central blasts and centrocytes. These pediatric type cases really have almost medium-sized, what was termed blastoid cells. So it didn't really fit, when you look carefully, it didn't really fit perfectly for central blasts or centrocytes. And to this time, we no longer grade or even think about grading pediatric type follicular lymphomas. So when you map this out in terms of age groups, we have sort of a two-phased bimodal distribution where the typical folliculars, as I mentioned, occur in older patients, whereas the pediatric type can occur, most commonly in the teenage adolescent years, but can occur up to, we've seen cases in patients 50, 60 years old. So when you have, so we begin to realize that this is an entity, and these patients basically, as the decades went on, people became, especially with the children, became more brave in terms of not treating them with chemotherapy, and the outcomes were the same. So over time, it began to be recognized that these patients really didn't need therapy. So now it became, to me, critical to sort of define these in a way, because it would prevent the unnecessary use of chemotherapy in young patients, which would potentially lead to other issues down the road. So we proposed the entity pediatric type follicular, characterized by a lack of BCL2, BCL6 arrangements and high proliferation fraction, and hypothesized that this was biologically distinct disease. But began to realize that was a major problem. So after this, the pediatric oncologists in our institution and then other related institutions would basically observe these patients and not treat them when they got seen by a pediatric oncologist. But when the patient was over 18 and got seen by an adult oncologist, they were getting treated still with R-CHOP. And the paper that we had, I don't think it was recognized. It wasn't really, there wasn't enough to define the as an entity. So we felt that there needed to be more work to really define, to pull this out as an entity. So we wanted to try to find the underlying biological distinction between these pediatric type cases from typical follicular lymphoma. So we collaborated with a large number of institutions, because these are uncommon lymphomas, to get these cases, and we sort of compared two types of cases: pediatric type folliculars, defined by the lack of BCL2 rearrangements and high proliferation fraction, and low stage typical folliculars. So these would be stage 1 to 2. So we're comparing apples and apples, but these either had a BCL2 arrangement, BCL6 arrangement, or a low proliferation fraction. And the idea was to really look at the mutational differences, genetic differences between these, to try to define what they were. So this was the cohort, and it had a similar elevated male to female ratio as we would expect. And again, these pediatric type cases were predominantly taken from the head and neck area. And the median age was younger for the pediatric type, median 14, whereas the median age for the pediatric. So I'm sorry. So this is breaking up the pediatric type into patients that were younger than 18 and older than 18. So we had both 16 younger and 10 older. And you can see the variations in therapy. So there was, most of the ones that were pediatric type were just excised as more and more recognized, whereas the low stage ones, there was much more frequency of radiation and chemotherapy. Follow-up was similar, and remission, no evidence of disease at last follow-up was uniformly occurring in the pediatric type, whereas the low stage, there was evidence of recurrence and progression in a significant number, and a transformation in almost 20% of the patients. So again, we had a similar sort of prognosis in terms of failure-free survival between the pediatric type and low stage follicular. So again, this was a representative cohort. So when we looked at these, one of the first things that we noticed was that the complexity of the genome by copy number alteration was really low in the pediatric type. So 0.5% of the genome altered by copy number alterations versus 10% of the genome in typical follicular lymphoma. And there was recurrent copy number neutral loss of heterozygosity at 1p36 in a significant 40% of cases, which has now been sort of standard, including TNF-RSF14, which we'll mention in a little bit. And what we found was, when we did targeted sequencing of a large number of genes, including all the genes, intentionally including all the genes that had ever been published related to follicular lymphoma, and found that the low stage conventional follicular lymphomas had a lot of the typical mutations published for follicular, including CREB-BP, EZH2, MLL2, whereas the pediatric type, whether they were younger than 18 or older than 18, almost none of them had these mutations, with the exception of TNF-RSF14, which is present, mutated across the board. So we were very excited, and we were ready to, jumping up and down and saying, hey, this is biological evidence that these are molecularly distinct. We had done whole exome, which looks at the whole, all the genes on 6 cases, and there were no prominent. We didn't note any prominent recurrent mutations at the time. And this is just a histogram showing the frequencies. So these are the most commonly mutated mutations in follicular, and you have two large cohorts of typical follicular. So you see the common players in follicular lymphoma, and you can see that for pediatric type, they're almost not present, with the exception of TNF RSF14. So at that time, we sort of were ready. We submitted this for publication, that this is really a genetically distinct, biologically distinct type of follicular lymphoma, and occurring in both children and adults with really good outcomes, maybe not needing therapy. Although we were still not sure what was driving these proliferations, so we submitted. And anecdotally, it's interesting. Basically, we had a reviewer who basically said, "Hey, we only looked at a certain number. How can you know that there's not something there?" And basically had us do exome in all the cases, which ended up being a blessing, because we found we had a relatively small number of cases. When we sequenced all of them, we found that 60% of them had mutations in genes of the MAP kinase pathway, especially MAP2K1. And you can see that it was a common mutation in the negative regulatory region of MAP2K1. And this particular mutation had also been previously described in melanoma and a subset of Langerhans histiocytosis, or Dorfman. So we were, thanks to that reviewer, we were very excited to sort of define a molecular event that could sort of separate these out as well from conventional follicular. And at the same time, there were several studies that basically were converged on top of each other, and to confirm this work. And this is a histogram sort of summarizing all of those papers. So you can see now that the pediatric type, well, in addition to not having the traditional mutation seen in conventional follicular, maintains mutations in TNF-RSF14, but also has unique mutations in the MAP kinase pathway, and as well as one of the publications showed that a subset had mutations in IRF8. So at this time, we were able to sort of confirm and define these as pediatric type of lymphoma, predominantly it was male predominance, limited stage disease with excellent prognosis, phenotypically showing expansive follicles with blastoid cells, a high proliferation fraction, a lack of the BCL2 and BCL6 arrangement, genetically pretty bland with the absence of the typical epigenetic modifier mutations with frequent MAP kinase pathway mutations. And the important point being that this entity is not defined by age, although it's more common in young adults, but has a unique biology that's shared by children and adults. And this became integral for the WHO 2016 classification, where this was then became a distinct entity, where unlike conventional follicular grading was no longer suggested. Importantly, it was clear that these really should be limited stage, and there should be no evidence of advanced stage disease, and there's no DLBCL component. And you can see that the blue box, or the sort of requirements for this, really are not only morphology and BCL2 negativity by stain, but also the lack of the BCL2, BCL6 rearrangements and that limited stage. So the clinical implications were that both children and adults with this disease really did, suggest that really did not require chemotherapy, and that MAP kinase mutations could be a marker for this disease. And so this particular patient that I presented initially was initially diagnosed as follicular lymphoma grade 3 of 3. This was a patient that would then, based on the new information, was re-diagnosed as pediatric type follicular at a different institution. And it's one that I remember fondly, because it was, as pathologists, we don't really interact with patients that much, but this was the first time a parent called, he contacted me and said, "Hey, I see it's this disease, and I see you suggest that, you don't need chemotherapy." And so I referred her to our oncologists, who are more familiar, who then, instead of getting R-CHOP, which was the initial planned therapy for this patient, basically was observed based on the recommendations. And to this day, it's been 5 years or so, 6. Patient is, the last time I heard, was doing really well. So it's one of the rewarding aspects of is when you could actually do something that impacts real patients. So this is another patient that I remember from a pathology point of view because a young patient again with cervical lymphadenopathy with follicular with follicular lymphoma. And you can see it has a sort of nodular appearance. um, Not so much a blastoid, but it you could maybe fix that. There was you can see CD21 positive meshworks and B cells. And BCL2 was clearly negative, and the proliferation fraction was more than 30%, relatively high. So this case I got this case in consultation, whether this could be a nice case of pediatric type follicular. My answer at the time was do the FISH for BCL2, BCL6, as I mentioned. That is part of the criteria. Um, and um, at the time, the I that the person sort of didn't really, I think the person was not pleased that I suggested that and really wanted to just call it, but did the FISH and found that while there was no BCL2 arrangement, there was a BCL6 arrangement. uh, so uh, Technically this would be a conventional focal pump that just was BCL2 negative. um, Again, I think clinically there was suspicion that this could still be pediatric type based on the BCL2 negativity. CT showed some axillary lymphadenopathy, which again doesn't fit for a pediatric type, and that was biopsied. And amazingly, that biopsy, instead of showing these large expanded follicles, showed um, uh, much smaller follicles with these eosinophilic deposits. And there were a lot of plasma cells there as well, and uh, 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 inclusions. Again, the CD21 was very tight, but in this case, the BCL2 was somewhat positive, and the proliferation fraction was much lower. So a lot of even the histology was more consistent for conventional uh, follicular lymphoma. So I mentioned this because as a lesson, that I always remind folks that it's not just the morphology of the BCL2 negativity, all the clinical features and the molecular, the BCL2, BCL6, and even looking at MUM1 are important to make this diagnosis. So in later years, we began looking at several other unique subtypes of follicular lymphoma. One of them being primary cutaneous follicular center lymphoma, which is a type of follicular lymphoma which has been well known over the years that it presents localized to skin lesions, and often in the head and trunk. And these lesions usually have recurrences, but dissemination to extracutaneous sites is pretty uncommon. And these patients have an excellent prognosis. And again, like pediatric type, have BCL2 negativity and lack BCL2 gene arrangements. Now, the differential for these cases is always, sort of skin localization or secondary involvement of the skin by conventional nodal follicular lymphoma, which are often BCL2 positive. So we were interested to see if these might have the same genetic features as pediatric type. So we looked at these cases, 40 cases of primary cutaneous follicular lymphoma, where we had follow-up and showed that there was persistent cutaneous restriction. Only a subset of them had subsequent systemic dissemination years later. And then there were 10 cases where we had secondary cutaneous follicular lymphoma defined by either nodal follicular lymphoma with subsequent cutaneous involvement, or cases where there was simultaneous presentation of cutaneous and nodal disease. And again, the hypothesis that these cases should be genetically distinct from typical follicular lymphoma, and what are the mutational differences. So again, there was not significant differences in the male to female ratio or in the median age group. The primary cutaneous ones did more often occur in the head, the scalp, face, shoulder, trunk, as we know. Whereas the ones with the secondary cutaneous involvement had a much more 40% of them were outside of this area. And then we had the four cases that were really presented as primary cutaneous follicular, we'll call that, but then later disseminated. And interestingly, all four of those cases were, initially present as would be expected in the scalp face. Um, when we looked at time to outcomes, we had pretty good initial remission, but a subset of these primary cutaneous cases had recurrence. Um, um, and these four cases, there were sort of a mix where there was, again, recurrence and disease at last follow-up. The time to recurrence was a little bit quicker was pretty quick for the ones that had subsequent dissemination. The ones that remained restricted to the skin took a longer time, but still recurred in the skin. One of the striking things that we noted, which I hadn't realized before this study, was that the recurrences were amazingly restricted to the same areas. So you can see here the primary location of the lesion, and the recurrence was almost always in the same spot, and even the second recurrence. Whereas the four patients that had initial primary cutaneous with subsequent dissemination had very distinct. One sort of went around in the skull, within the skull and the brain. Another one went to bone marrow or breast. So definitely a biological difference in sort of how these, not only in terms of um, being outside the skin, but very wide behavioral differences. It seems to be that these cutaneous cases, were really localized, and they didn't have the ability to really escape that area. And so when we did the genetic analysis, we found that the secondary cutaneous lymphomas, as would be expected, were very commonly involved the traditional mutations in the epigenetic genes like CREB-BP, KM2D, and those mutations. And we only were able to do, of the four cases that subsequently disseminated, only two of them we were able to analyze, and basically they also had common mutations in those genes. Now, the primary cutaneous ones certainly had less involvement of those of CREB-BP and KMT2D, but there were still a subset of cases that had one or more, a subset, rarely occurred. So it wasn't as clean and as specific as we found in pediatric type, but definitely there was a difference in the overall burden of mutations and the distribution. So we came up with a algorithm um, to sort of to use to help sort of predict whether a case would be, at the time of diagnosis, it might look like primary cutaneous follicular center, but to predict the cases that might have future dissemination. And basically, the algorithm was having two or more of the following criteria, either having KMT2D and CREB-BP, which seemed an important common feature of the cases, greater than 3 chromatin modifier variants, so some of the other genes that were chromatin modifier, and then the presence or absence of BCL2 gene arrangement. So if you had two of these, then it seemed very highly correlative with, even if it looked like pediatric type, that could later have systemic disease. And a couple of example cases that we tested on this: a 74-year-old male with a scalp lesion, no evidence of systemic disease. The morphology was pretty good for primary cutaneous. They're usually larger with sort of large centrocyte-like appearance. The immunohistochemistry was favorable for that in terms of it had the high proliferation fraction. It had lack of BCL2 expression, but you can see in the skin there's CD10 and BCL6, which are those neoplastic cells. And commonly we see sort of a hollow CD21 meshwork, which we're seeing here. So this patient didn't have CREB-BP or KMT2D or any of the other epigenetic modifier or BCL2, and it was signed out as possible primary cutaneous. Oh, sorry. So yeah, so this was signed out as primary cutaneous and basically did not relapse or recur. A different patient here is a 47-year-old man with a forehead lesion, discrete nodular infiltrate. CD21 meshworks again were weak. No BCL2, BCL6 rearrangements. Again, this was signed out as possible cutaneous, primary cutaneous follicle center, but alternatively, this had a CREB-EP and KTM2D mutation as well as EZH2. Um, and then imaging later showed concurrent nodal involvement. Another patient with BCL2 positive disease in the scalp, CD21 meshworks are present. There was a BCL2 gene arrangement. It was still signed out as primary cutaneous follicle center, but again, this had the CREB-EP, KTM2D, and BCL2 gene arrangement. So the algorithm sort of was consistent with what we would expect here. This really needs to be further, evaluated with more cases. Yes, and this last patient later, there was no concurrent nodal involvement by imaging, but this patient later had bone marrow disease. So this is sort of some current work we're doing that I've added. We're looking at patients with Hashimoto's with follicular lymphoma, and we're finding that there is common mutations in TNF14 as well as PDL1 and TET2 mutations. And interestingly, this may have some role in the interaction between T cells and lymphoma cells, which involve these receptors, TNF14 and PDL1. So this is work in progress. So just to summarize what I've talked about so far, we have, conventional, we've been able basically by identifying unique subsets of these, unique features within this disease entity, we've been able to carve out different subsets that have, I think, important impact on how these diseases behave. One is the typical follicular lymphoma where you have BCL2, BCL6 gene arrangements, and you have the typical CREB-EP, KM2D, and epigenetic modifier mutations with low proliferation fraction. These we think of as starting in the bone marrow with the BCL2 gene rearrangements, and then slowly acquiring mutations until they present as follicular in the 6th decade in that area. The pediatric type follicular basically lacks the BCL2, BCL6 rearrangements with no CREB-EP mutations or KMT2D mutations, but they have a high frequency of MAP kinase mutations. So there seems to be their morphological features as well as genetic features that make them distinct. We think of these as locally stimulated clonal follicular proliferations. They never progress or go to other sites. So most likely there's some kind of local antigen stimulation that's driving a process. So it's probably very different than the conventional, which starts in the bone marrow with a BCL2 gene arrangement. And then primary cutaneous follicle center lymphoma mostly has no rearrangements or some of these epigenetic modifier mutations, and they present in the skin with high proliferation fraction. Also, the majority of these, therefore, are probably locally stimulated, antigen-stimulated processes in the skin that hasn't been proven yet. But a subset, we have to be careful because a subset of these may mimic that kind of biology, but actually harbor these mutations and maybe then will later present as more conventional, more disseminated later on. So from there, after doing a lot of this work, began to realize that there's only so much that one can do looking retrospectively at patient samples. We can characterize mutations and make correlate with behavior, but what we can't do is ask experimental questions. We can't do things like in many ways like single cell RNA-seq or flow cytometry or modulate some aspect of the disease to ask a hypothetical question. So to do that, we need a model, and there are genetic models of things like DLBCL, but usually those are based on very limited genetic alterations. Now, lymphomas, as I've shown many of times, that these lymphomas have numerous mutations, and so these genetic models don't really match exactly the patient, and they're usually a model of all. Like if you have a model of the DLBCL genetic model, it's a model of all these. So you can ask very high level questions around the biology of lymphoma, but if you really want to get at what is the difference between patient A and patient B with the LBCL, and why is this patient responding to R-CHOP and the other patient not responding to R-CHOP, it's very hard to do, and just looking at mutations doesn't get there. So what we've talked about in the first half have been very rare subsets of follicular lymphoma with unique genetic features, but the rest of the follicular lymphomas, basically there's a lot of variability, and some patients don't require any therapy. Some patients require therapy and relapse really early and do poorly. And we have no real way to predict that for follicular. And so we know in that example that the rest of those follicular lymphomas have very similar mutational profiles, although different, but the common ones are very similar. So I began to be very interested in trying to develop a better model. And so what we focused on was patient-derived xenograft models, and which basically means taking a patient's sample and putting it in an immunodeficient mouse and allowing that to grow. And what the advantage of that is that you are capturing immediately capturing the genetic variability of each patient. And you could actually, if you can maintain those features, you could actually look at the differences between each patient, right? And really get to sort of patient defined therapies. So the start of this was developing a biorepository at MGH, where we basically have a pipeline. So at MGH, every lymphoma goes to the frozen lab and gets triaged. And at that time, if the tissue is, if there's sufficient tissue to do the clinical work as well as extra tissue, we will bank some of that tissue and do it in a viably frozen way. So not snap frozen, viably frozen, which allows us to do things like making the PDXs, doing flow cytometry, doing anything you really want. We've been doing that for probably seven years now, but more, it's more robust now, and we do it in a very objective way. So there are investigators who basically have their lymphoma of interest and identify patients with that lymphoma, and then go that way. We bank everything we can, because the rare cases, the rare entities, it's really hard to do it if you're looking for them. But if you're just collecting, we'll get we'll get some reactive things, but we'll also get sort of the rare lymphomas because they just show up. So that pipeline has been very helpful for us. Now, making a xenograft, this is just an image showing what we do. So we'll take one of those viably frozen pieces of tissue, which is basically in this circle, this size, it's like a rice grain. We'll initially insert this into the renal capsule of the kidney. So we'll make a little incision, take the kidney out, make a little incision in the renal capsule, slide the tissue underneath the renal capsule, close that, and then put the kidney back, close the mouse. Depending on how aggressive it is, it could be 2 weeks, it could be several months. We'll generally get a tumor, and this is just showing the development of the tumor over a period of time. And so I'm going to we applied this to really make a difference, I think, in a subset of patients with aggressive lymphoma, which is where we've been focusing. So again, I'll just present the index case. 42-year-old man presented to MGH with night sweats, anemia, tons of adenopathy, splenic lesions, and then a follow-up was obtained. And so this patient ended up having this basically large cell lymphoma. You can sort of see there's sheets of large cells in this case. Has more of a plasma blastic appearance, or a little bit of a plasmablastic appearance. You can see that this cell stained for ALK, but negative for CD20 and CD3, has some CD8. So this is classic for ALK, large B cell lymphoma, which was reported in 1997 for the first time. These are rare, really aggressive lymphomas that have more of a plasmablast sort of feature. They lack B cell and T cell markers. The patients are often pretty young, the median age 43, but the patients have a dismal prognosis. They usually die within a few years. As a result, because they're aggressive, there's been no ability to do clinical trials. These are driven by ALK fusion protein, so anaplastic lymphoma kinase, which is a receptor tyrosine kinase discovered in 1994. And these fusions in ALK positive large B-cell lymphoma, the partner is usually clathrin, which is a structural protein, whereas ALK positive ALCL, which is a T-cell lymphoma, also has ALK fusions, but they have different partners, most commonly MPM. And downstream of this activated ALK, you activate a number of signaling pathways, including JAK-STAT, PI3 kinase pathways, ERK. And you can see that basically, particularly patients with uh, uh, advanced stage disease, the prognosis is really dismal. And so there have been you know, the discovery and utility of ALK inhibitors, which actually inhibit the activity of the ALK fusion proteins. And these are present in lung cancers, as you know, and other kinds of cancers. They've been used very successfully. And there have been a number of generations in terms of improving the efficacy of these. So there's the first generation, second, third generation. So clearly, one would think, why not try these in these patients? These patients generally get several therapies, very aggressive chemotherapy regimens, and then they fail those. Anecdotally, there had been attempts to use crizotinib, the first generation ALK inhibitor, for these patients, but these basically really did not work. These patients failed that, and this is the index patient I mentioned. This is the course that he got EPOC, DHAP, had crizotinib with progressive disease, lenalidomide, a whole set of things, and basically all these failed. So the rationale for PDX modeling is basically if you generate these models, and actually when you generate them, you can keep generating. So in other words, the tumor that comes from the first PDX, you can make multiple seeds that you can freeze down, make subsequent passages, and essentially grow them without, and you can do essentially a little clinical trials with these mice. And so this is just another demonstration of the creation of the PDX here. We use, in terms of detecting the tumor when it's generated, it's hard to do when we do the renal. So we use ultrasound, and I did include this just to show what that looks like. So this is a normal kidney, this little horseshoe-shaped structure here, that's normal. But then over here, you'll see this is a kidney with a tumor in it, and you can see the horseshoe-shaped kidney structure, but then you can see this lesion here, that's the tumor. So that's how we know when we have a tumor. And this was our first PDX of the ALK. So you can see this is the PDX, the kidney and the tumor in the kidney. On the bottom, you can see a normal size of the mouse kidney, and then a kidney replaced by tumor after about a month and a half. And you can see that the histology of the patient and the PDX is pretty similar. The PDX also showing the ALK expression and the lack of 3 and 20, and genetically, essentially identical. So we basically implant, we do an engraftment check, and we check a couple more times to make sure that the tumors are growing, and then we sort of match select by tumor size and growth of vehicle treatment and control groups, and then we follow with ultrasound to look at growth. This is showing at the beginning the ultrasound of the initial at the time of start of the drug. You can see that the tumor here is, they both have really large tumors. And then this was at the time of sacrifice. So you can see that the vehicle treated mice had a ton of tumor in the kidney, whereas this is the lorlatinib treated mouse really responded. And I should say, what we had done in this experiment was we treated the mice with first generation, second generation, third generation, lorlatinib being advanced generation ALK inhibitor, to see if there was a difference. You can see by Western that the ALK inhibition really was on target, and you can see with the lorlatinib, the second generation, you can see that there was a loss of phosphorylation of ALK and downstream things like STAT, phosphorylated STAT, and SHP2. You can see that the difference at day 0, you can see they're both growing before, and then the lorlatinib treated mice really responded by 2 weeks. The tumors were gone. So what was really exciting was when we compared lorlatinib and the first generation crizotinib, you can see that the purple are the vehicle. They are growing. All these mice are growing. Each of the this is measuring tumor volume in all of these mice. You can see that at the time of treatment, the vehicle treated mice continue to grow. The crizotinib treated mice have a sort of a lag. There's a response, and then they grow, and that's essentially what we see in patients. There's an initial response, and then they continue to grow. But then the lorlatinib treated mice really had a persistent response. And we also did this with alectinib, which is a third generation ALK inhibitor. So this really demonstrated that there was activity with second and third generation ALK inhibitors. I collaborated with my colleague oncologist at MGH, and we identified patients across the country at Sloan Kettering and other locations who had this disease and basically had really grim prognosis. And we suggested they try the lorlatinib therapy, and essentially all of the patients responded. One of our initial patients basically so the other things that we realized once patient went into remission, you quickly have to follow that up with a transplant. And so one of the patients, the initial patient, basically succumbed to complications of GVHD therapy, which is an issue. But of the patients, we had one with progressive disease, but two of the patients basically are in a long-term clinical remission, several years out. And this is just an imaging example of one of the patients where the patient came in, one of the more recent patients who came in with disease, was basically told to make arrangements, and then was treated with lorlatinib. After a couple months, no evidence of disease. So this image is just to show the similarities between the ALK large B-cell lymphoma and another subtype, which is plasmablastic lymphoma, which is driven by or associated with Epstein-Barr virus. So very different biologies, but really they both have a plasmablastic picture and an identical clinical outcome. So I call these plasmablastic type lymphomas. Actually, they were both first reported in 1997. They're both rare. They both have dismal prognoses. They both have a plasmablastic phenotype. And I think that if we think about cell of origin, really a lot of B-cell lymphomas derive from many different points of this sort of developmental pathway. But I think the plasmablastic lymphomas and the ALK large B-cell lymphomas arise from a very transient phase called plasmablasts, which they're between mature B cells and plasma cells. They don't survive. They're not present very long, and I think that's part of the reason why these are so rare. So some of the questions that we're looking to follow up with are what really mediates the crizotinib failures, which we're beginning to think is a pump that pumps the drug out of the cell, and what mediates the difference between the very poor prognosis of ALK positive large B cell lymphoma versus ALK positive T cell lymphomas, which actually have a not too bad prognosis, and looking at the biology of these different lymphomas. So we're working now on really characterizing these models. And so when we generate the patient tumor, you can generate first generation mice, PDX mice, and then second generation. And we're trying to characterize the genetics of these and the RNA expression of these. And the other thing that we're able to do now is take these PDXs and grow them in culture as organoids. And that allows us to do high throughput drug screens on these models. And so this is just showing this is just showing the how identical sort of a PDX and a patient are. So this showing all the mutations and how similar they are, and even the allele frequencies have a linear relationship between the patient and the tumor. So genetically, these are very similar, and the idea is that these cultures are not cell lines, and that they don't have some transformative event, but they're primary cultures, so they maintain the genetics of the original patient tumor. And we're now using 10x technology, a new version of this that allows you to do this on formalin fixed tissue to characterize these models. And you can see that we can actually, using single cell seq, we can characterize the populations in the patient tumor and the PDX. So here we can see neoplastic cells and T cells, fibroblasts, macrophages, and endothelial cells. And in the primary PDX from the same patient, we characterize a similar population of neoplastic cells and T cells, which are characterizing further as well as the fibroblasts, whereas the macrophages and endothelial cells, as would be expected, don't persist in the PDX. And then subsequently, you begin to lose some of the T cell populations, but the fibroblast population is still there. So we're looking to see anything special about the fibroblasts. When you superimpose these, you can see that between the patient PDX and the patient tumor, you can see there's a lot of overlap between these populations, but seems like the T cell population from the patient and from the PDX are different. So we're looking at that as well as expression differences between tumors, between patients, and between patient and PDXs. I know we have a little bit of time, but one of the things we're looking to expand to all DLBCL. So these large cell lymphomas I talked about are sort of less than 1% of DLBCLs, but DLBCLs overall are a large fraction of lymphomas. And as you may know, these lymphomas are now retrospectively have been molecularly characterized into multiple molecular subgroups with very distinct overall survival differences. So these are predictive, and already based on the mutational profile, there are candidate drug targets that can be considered. There are multiple of these classifications. So we have actually in our bank a large number, about 50 patients with DLBCL that are well characterized, and we think that these represent the molecular subtypes of DLBCL well. So our goal is to basically generate a library of these lymphoma subtypes, do high throughput screens on them in the organoid system, then test these out in the mice, and really, go beyond retrospective analyses of lymphomas, but really begin to characterize potentially how these mutations can predict the response and what therapies would work. So, in conclusion, I talked a little about the genetic differences between these different follicular lymphomas, talked about some of our work with ALK and the plasmablastic-type lymphomas, and tried to illustrate the potential of the PDX models to inform how we can use them to inform therapeutic options for patients with rare malignancies that are hard to study otherwise, but also potentially to expand these to look at, large cell lymphoma in general and how we can understand the molecular subtypes. So with that, I thank you, and again, I really appreciate the invitation to share our work. And if there are time for questions, I'm happy to answer.
Thank you, Abner. I really, really enjoyed your presentation. I know there are questions in the field. I was going to ask a first question, if that's okay. I was curious for your PDX model. The first question I had is, how well do low-grade lymphomas grow? And in the lymphomas that do grow, how often do you see murine cells kind of intercalated in that model?
Yeah, those are great questions. So there was a whole other part of my talk that I thankfully deleted, which was around follicular lymphoma. And they are much harder to grow. But when you get them to grow, the exciting thing is you can get because those tend to have a microenvironment, and you can get the microenvironmental cells, like the T cells, and even some of the myeloid cells to grow in the model for some period of time. But the engraftment rate for like a DLBCL is essentially 100%, but for follicular lymphoma, it's like a fraction of that. So they're much harder to grow, but what we're trying to do now is develop an in vitro organoid model. So if we can grow them as a PDX and then develop and then do most of the work as an in vitro organoid model, I think that's going to be the way to leverage them. In all of these models, you do have a small subset of murine cells, but usually they're stromal cells or endothelial cells. So it's a very small, I would say less than 2% of the population usually.
Great. Looks like Dr. Beckman had a question.
Hi, my name is Amy Beckman. I really enjoyed your talk. Thank you so much for that. I have actually two questions for you. First of all, about pediatric follicular lymphoma. Do you think, whenever these cases are so rare, but when I've seen them, I've thought, maybe we should not be calling these a lymphoma. And do you think we're at the point where these are well enough defined that we could call them something else, like a lymphoproliferative disorder? And then my second question, if you have time, is if you could talk a little bit about pediatric type nodal marginal zone lymphoma and how that relates to follicular lymphoma.
Yeah, so I have interesting anecdotes on both. So for the pediatric, for the first question, the nomenclature, when we did our work and it became was going to be part of the the new entity, there was a lot of debate about what to call them. And so one half of the consensus was, let's call them because it was realized that these are not really lymphomas. So can we call them lymphoproliferative disorder of unknown uh, significance? Yeah.
Significance, yeah.
Yeah, and so the feedback was that makes sense, but patients getting that diagnosis would probably be more afraid if we called it anything with unknown significance versus a defined entity. So we decided, and I think the rationale for calling it to crossing over to lymphoma is essentially that it's one of the only entities that I know of where you have like a effacement of the architecture by a clonal population. So therefore, that fulfills lymphoma. There are not many cases of that are sort of benign, although they're more and more commonly. I think there are more of them that are commonly recognized now. So I think I sort of sometimes joke and say this is like sort of the adenoma of the heme world. And that it is a neoplastic process, or at least on some level, but it's whether it should be considered a malignant process. Definitely not a malignant process, and whether we should use the term lymphoma is questionable. So I think your question is well taken. In terms of the pediatric nodal marginal zone, so when we first characterized these, we also looked at some pediatric nodal marginal zone, and there are actually these unusual cases. You might have seen some where there are some features of both. So they may look like a follicular pattern, but they have CD10, but they may not have some of the features of follicular. And we proposed that these could be a spectrum. At the time, that idea was rejected, but very recently, there's now data showing that a lot of those pediatric nodal marginal zones share the genetic alterations of pediatric type follicular. And so I think the idea that these are a spectrum, probably a spectrum of the same sort of antigen stimulated proliferation, that's, I think, more and more accepted. So at one time it was thought that these were, hardcore different, but I think now I really see them as the biology being a spectrum.
Great. Well, we probably have time for just one quick question from Dr. Yohe.
I don't know if it's going to be quick, but so I'm Dr. Yohe. Thank you for the talk. I do both heme path and I also direct our molecular lab here. And so my question has to do with what sort of testing you guys are doing from a clinical standpoint for lymphomas and whether or not you're actually getting reimbursed for any of it.
Yeah, that's the classic. That's a classic question. Yeah. So we are not doing sort of uniform molecular testing on all our lymphomas, but we do a lot more of that. A lot of our stuff we do either internally with a internal platform that we use for our myeloid cases because the cases overlap.
Yep.
And I believe we're getting reimbursed for some of them. There are some entities where we can argue, but we don't do it uniformly because we don't get reimbursed. We also send a lot of stuff to Boston Gene, which is a company that does exome and sequencing, and they have a system which is nice where a lot of them get reimbursed, but if they don't get reimbursed, they sort of eat that cost. So it allows us to do it. It's really disappointing, as you know, because I think on the myeloid side, there's a panel of myeloid markers that are reimbursed, and then they also have other markers, and then you learn things, and then people report them, and then you can add more things. But we don't have that snowball started yet, where we can't learn anything if we can't do it because we can't reimburse. But we're trying.
Thanks.
Thank you so much for those questions. Hopefully they address them. Okay.
Thank you so much for speaking today, Dr. Louissaint. I really enjoyed your presentation. I learned a lot.
Thanks for the invitation. I really appreciate it.